So, Norm. Okay. You want to get started, Norm? Hello? Yes?
I just had to unmute myself. Sorry. Yes.
Yes,
Let's get
please.
started.
Okay. I'm pleased to introduce my colleague, Adam Fields. Adam is a translational neuroscientist who studies molecular mechanisms involved in neurodegeneration in the context of HIV infection. He earned his PhD in biomedical sciences from the University of North Texas, and in 2012 joined the Akeem lab here at UCSD. Adam is now an assistant professor of psychiatry and associate director of the neurobiology unit of the HNRC. And his talk today is entitled Mitochondrial Alterations in HIV and ART-associated Neurotoxicity. Thank you, Adam.
Oh, thank you, Bob. Just add, I don't mean to correct you, but before I was in Eliza Messia's lab from 2012 to 2016, and then a King lab the last year. Just to give credit where credit is due.
Okay.
Anyway, all right, thanks for having me today. I've really enjoyed this platform. I don't know, we've been doing it for like 2 years almost now, and it's been great. In lieu of all the lack of in-person conferences, it's just been great to keep on hearing all the good science. Sorry about that. Well, we can jump right to this. Y'all just heard the title anyway. But so I kept the title pretty generic because we're going to be talking. I wasn't exactly sure what I would present, but we're really covering the arc of things that of studies that we've done over the past about 6 years. So just to give you an overview of what we'll talk about, I'll tell you a little bit about experimental neuropathology, the work that we do in the different models and specimens we look at. And then I know I'm preaching to the choir, so I'll briefly just talk about why we're looking at the neurological disorders, a little bit about why we think mitochondrial biology and the aspects of mitochondrial biology that we think are important. And then what we've gotten into the last couple of years is the interplay between different cells in the brain. And so then we'll break down the findings into 3 sections. The first one will be HIV and mitochondrial dynamics, mitochondrial fission and fusion, a little bit about mitophagy. And then we'll look at mitochondrial biogenesis, or the generation of mitochondria in neurons, but also in glial cells, mainly astroglia. And then finally, we'll finish up talking about how astroglial role in HIV and ART-induced mitochondrial dysfunction. We'll briefly touch on future directions, and of course, acknowledge all the folks that have helped me throughout this time. And so here on the right, you'll see these little pictures, and some of the data will come from human brain tissue. Some of it's going to come from animal models, and some from in vitro models of brain cells. So for each slide where there's data, you'll see one of these pictures in the right-hand corner. So it'll kind of keep us grounded on what kind of data we're looking at, because we're kind of jumping back and forth. So let's see. Okay, went to the right one. These things are very finicky, I feel like, when it's on presenter mode. Okay, so experimental neuropathology. What do we mean by that? Basically, everything we do is for the humans. So we start with human disease, and we get our brain tissues from mainly the ones that I'll show today from the National NeuroAIDS Tissue Consortium. So you'll see this signal or this little picture of a brain here, and we'll do molecular, neuropathological, look at biochemical. We've got some transcriptomic analysis of brain tissue, and then we translate that to look for the mechanisms underlying the patterns that we find in the human brain tissue. We use these in vivo and in vitro models. And the ultimate goal is to come up with therapeutic strategies to target the mechanisms, and then ultimately get into clinical trials. And we haven't gotten that far with the central nervous system, but we do have a clinical trial going. I'm not 100% sure if it's funded and going yet, but for peripheral neuropathy to target mitochondrial biogenesis. I won't talk about that today, but it's something I'm pretty proud of. I couldn't have done it without obviously a lot of help from other people, but it was nice to be a part of it, to see something come from the bench and is actually getting to the clinic. So I know most of you know this, but sort of just in case, still a lot of people infected with HIV, despite the advent of antiretroviral therapy and its wide use and effectiveness. Still people have lots of, or there's a lot of people with neurocognitive disorders, but also other mood disorders. There's some overlap with Alzheimer's disease. We won't really talk about that today, but I think it's important as these people are living longer and aging, as I know many of you do. And then a large proportion still have sensory neuropathy, which it can be really devastating for daily functioning and just doing things that we all take for granted. I won't talk about it today unless somehow I get through all this early. I think I do have some at the end, but I doubt we'll get there. So just to recap, I know you all know this, but still the model that we use, or the idea, is that HIV gets into the brain very early after infection by immune cells across the blood-brain barrier, HIV-infected immune cells. And once in the brain, they can release HIV proteins and cytokines. They also infect resident microglia. There's some evidence they infect astrocytes. And the HIV proteins and the virus can also affect, not infect, but affect neurons and processes in neurons. And then you also have antiretroviral therapy, which we'll talk about today, how that can have deleterious effects, not only on neurons, but also astroglia. So mainly what we'll talk about today is what happens with mitochondria and neurons in the brains and some possible mechanisms for that. And then also astrocytes and how they play into it. And sort of a theme that it's sort of formed in, as I've been doing this over the years, is neurons, definitely mitochondria. You know, there's neuronal dysfunction, there is some neuronal damage, and the mitochondria, as I'll show you, are damaged in the neurons. And I'm sure there's many pathways to that. But I think based on what I've seen and others, and I'll mention the other groups too, that you're getting the sort of patterns of mitochondrial pathology that you see. It appears to be, in some ways, a generalized response to stress by the neurons, and also in astrocytes, but they react a little bit differently as far as their metabolism. We'll get into that, but just kind of wanted to point that out at the beginning. And I'm sure everyone has seen this many times, but I like to just remind people that the neurocognitive disorders remain a problem, although in the CART era, as you see on the right, the severe forms are not as prevalent, but the less severe forms have increased in prevalence. And then there's, of course, are the other mood disorders, and then also peripheral neuropathies and peripheral neuronal dysfunction. That still needs to be addressed. We still need to figure out the mechanism. We still need to provide therapeutics for these people. So mitochondria, a lot of people, when they hear about mitochondria, they think about 8th grade biology, and they're the powerhouse of the cell, which is completely true. Much of the energy for the cell is generated in mitochondria, but there's many other things that you need, processes for which you need healthy mitochondria. So they produce metabolites for cell signaling, gene expression, so epigenetic regulation of the DNA, building blocks for larger macromolecules, buffering of ions like calcium, and then they regulate apoptosis. And they really aren't these circular or ovals that you see in the textbooks. They exist in, I kind of like to describe it, maybe you've seen a lava lamp. I don't think they're as popular as they once were, but they bleb off, they're generated, or around the nucleus, the mitochondrial associated membrane with the endoplasmic reticulum, and that's sort of where the genesis happens. or at least like that's the origination of them, but they bleb off, and it's a network of these of mitochondria that they can then pinch apart in a process called fission, and they can fuse together with other mitochondria. And especially in neurons, it's important because you need mitochondria and energy to extend in cellular processes. So you can imagine there's lots of points of mitochondrial biology that could be disrupted and prevent this network from functioning properly, aside from all the metabolites and buffering and cell signaling that they participate in. So we'll talk more about that, but I just kind of wanted to set the stage just in case you were thinking about the circles in the cell, which you really need to draw diagrams like this, which covers the process or sort of life cycle of mitochondria, from mitochondrial biogenesis to the dynamics, which would be the fusion of mitochondria, to increase mitochondrial size, to the fission, pinching apart of mitochondria, so that maybe they need to be transported to different parts of the cell along microtubules, or damaged mitochondria need to be recycled via mitophagy. And here you'll see a legend with different HIV proteins and ART drugs indicated. And throughout this process, you'll see that it's been published, but not just me, many folks have investigated this. Different HIV proteins and ART drugs interfere with these different processes. Today, we're really going to talk about mitochondrial biogenesis, which is initiated whenever you have low levels of energy. So high AMP, low ATP will activate an AMP kinase, which will activate transcriptional machinery, starting with PGC1 alpha and some other transcription factors we won't talk about today. And that trend is translocated to the nucleus, where you get massive gene expression, and one protein or another transcription factor that is expressed is TFAM, transcription factor at mitochondria. And it is translocated to the mitochondria, where it attaches to mitochondrial DNA, and it facilitates replication of the DNA, but also gene expression from the mitochondrial DNA. And you need nuclear gene expression and also mitochondrial DNA gene expression to produce new mitochondria, which you could imagine would be important for generating new synapses, learning, forming memories. And then you have dynamics, as I mentioned, the fusion and fission, and we're going to talk about how that's disrupted. And again, it is what it sounds like. You're bringing multiple mitochondria together in a process of fusion to make a larger mitochondria. And in fission, you're pinching mitochondria apart in order to recycle them or to transport them to the different parts of the cell. I want to come back and mention, I should have mentioned mitofusin 1 and 2, or GTPases. These are enzymes that facilitate the fusion. And DNM1L is another GTPase that facilitates the fission. And I just want to say those now because we will be talking about them later. And so I mentioned the interplay between neurons and astroglia. For good reason. Most studies of neurodegenerative diseases have focused on neurons, and mitochondrial function in these, or dysfunction in these diseases, have focused on the function in neurons. But other cells have need energy. They also use mitochondria. And astrocytes are particularly important, as you may or may not know, that they form part of the blood-brain barrier. They transport glucose from the peripheral blood supply, the systemic blood supply, into the brain. They'll break down glucose through glycolysis, but in general, or at least resting state, it's believed that they don't use lactate, or they generate lactate, but they don't metabolize that to pyruvate and then use mitochondria as a major source of energy. Instead, they'll transport the lactate into the extracellular space, where it's taken up by neurons, and then the neurons use mitochondria to generate the energy they need. Astrocytes, sorry, I went too fast. Astrocytes also recycle glutamate, which is important for regulating synaptic transmission. And so I just want to get the point across that if astrocytes are dysfunctional, or if neurons are to function correctly, astrocytes need to be properly functioning. functioning properly. And in the brain, in HIV proteins and ART drugs, they have profound effects on astrocytes, which we'll talk about, which it makes sense to me, at least, that would compromise the metabolism in neurons, but maybe I can convince you of that. But other people have already shown that in Alzheimer's disease, these processes are disrupted, and in other neurodegenerative diseases, and more commonly, my folks like Kathleen Borgman and Diane Langford, Valeria Avicina and Attalo Machete, they've really looked at maybe not as much the latter as the interplay, but the former two have really looked at the interplay between astrocytes and neurons and drugs of abuse, actually. So it's becoming, we're becoming more aware of it as a field. So in astrocytes, much like microglia, which I know are very popular for good reason, they exist on in a continuum of states. It's not as binary as A1 to A2, but at the ends, you could characterize them as pro-reactive or reactive astrocytes, pro-inflammatory, and on the other end of the continuum, more homeostatic, and probably providing the lactate and the neurotrophic factors to the neurons. And during disease or some sort of insult, you probably need some A1 reactive astrocytes to rectify the situation. However, as time goes on, I think the system was meant so that the reactive astrocytes sort of skew back to the A2 astrocytes, where they are more homeostatic and can care more for the neurons instead of getting rid of the insults, whatever they may be. So really what we're talking about and what we think is there's probably a preponderance of A1 astrocytes. And if we could get it back to more A2 astrocytes, that'd probably be good for the brain. Okay, so first we're going to talk about fusion and fission and dynamics. So I put these green boxes here. And we'll talk about how we do this. As I said, we get a lot of brain specimens from the National NeuroAIDS Tissue Consortium, and we will stratify them by cognitive diagnoses. We're beginning to work on things like depression, but this first work was, I think, probably performed back in 2014, 2013, something like that. Stratify them by these different diagnoses of cognitive impairment. We look at particular regions of the brain. In this case, we're looking at the frontal cortex, Brodmann area 46, which is implicated in executive function. And then we take a multifactorial approach at analyzing the pathogenic patterns in these brain. We'll look at protein levels by immunoblot. We'll look at the localization of the proteins in the different cell types by immunohistochemistry, confocal microscopy. We'll look at the mitochondrial DNA quality, then we'll look at gene expression of other mRNAs through real-time PCR, and then we also do some transcriptomics analysis that I'll talk about in a little bit. So we basically hypothesized that mitochondrial dynamics could be altered in these brain tissues. And this really came about because over about the last 15 years, the enzymes that control mitochondrial dynamics were identified. So we were able to look into this process. And what we found, as you can see pretty well with this Western blot, if you go left to right, you have brain protein from neurocognitively unimpaired, and then sort of increasing severity of the diagnoses from ANI, MND to HAD. But DNM1L is that GTPase. It's an enzyme that facilitated the fission. And you see a large, robust band in these brains, but as you move left to right, you'll see that you see less and less. And we have repeated this. Every time I look at new brains, I always look at DNM1L to sort of confirm this, and something we've seen over and over. The activated form of DNM1L is also reduced from left to right. Mitofusin-1, which is an enzyme that facilitates the fusion of the mitochondria to make a larger mitochondria, it was not significantly different between the brain regions, although you could see a slight increase. We normalized all this to actin and quantified it. And on the y-axis, you'll see DNM1L to actin. And you can see that you do see a decrease in the most severe cases compared to the cognitively unimpaired and even to ANI. The same goes for the activated form, and the MFN1, while again, it's not significant, you do see a slight increase. So all this suggests that you would have larger mitochondria because you'd have less mitochondrial fission. So we wanted to see, well, is this happening in neurons? And you can't see that from a Western blot, because a Western blot, you're really just taking a piece of brain, chopping it up, running it on a gel and seeing total protein. So what you can do is look at immunohistochemistry, use antibodies on these same brains. We take very thin sections and we incubate them with antibodies against these same proteins, and then we can localize and see what kind of cells they're in. And what you'll see in this top panel is DNM1L signal in the control brains. You see these pyramidal shaped neurons in the cortex, and you see a very dark staining for DNM1L. But in the hand brains, you have less staining. And when you quantify, you see a significant reduction in this DNM1L. Mitofusin, you had an opposite effect. You had not as much mitofusin in the control brains, but you see the signal is much more intense in the hand brains. And this was somewhat of a surprise that it was so robust, but sort of getting back to the general hypothesis that we're sort of forming is mitochondrial fusion is in other diseases and in vitro models, it's been seen as a sort of a compensatory mechanism against stress. And the neuron is maybe sort of calling the troops back and pulling the mitochondria back until things settle down. And those things could be like HIV infection, chronic activation of astrocytes or microglia. And that could be what happened. But we're going to show some more stuff here. I just kind of want to plant that seed. We also looked to sort of confirm that this is affecting mitochondria. We looked at TOM40, which is a mitochondrial membrane protein, and you can use the antibodies to identify the localization and get an approximate size for the mitochondria. And so in the control brains, you see that the TOM40 signal is fairly dispersed throughout the neuron. However, in the hand brains, you see a larger and more punctate TOM40 signal. And when we measured the average size of these dark punctae, you do see an increased size of these dark punctae in the hand brain. So all this is suggesting that mitochondrial fission could be reduced, mitochondrial fusion is increased. So we use confocal microscopy and those same antibodies and double immunolabeling. So we take the same tissue of the same slice of tissue and we incubate it with two antibodies. One for DNM1L, you can see right here. Hope you all can see my cursor because I'm using it. So DNM1L antibody and TOM40 antibody. DNM1L is your green signal. TOM40, which is indicating the mitochondria, in theory at least, is the red signal. So where you have red and green, you'll see yellow. And in the control brains, you see lots of yellow signal. But in the hand brains, you saw less yellow signal, indicating so there's less DNM1L co-localizing with mitochondria. And you see a significant reduction in the DNM1L co-localizing with mitochondria in the hand brain, suggesting again that there's less mitochondrial fission. And then we wanted to get a closer look. So we used electron microscopy. We slice these brains very, very thin. And then you coat them with some metal so that ions or electrons will bounce off of them or pass through them. And you're able to get images at an ultrastructural level of the mitochondria. And hopefully you can see that in the control brains, you have nice cristae and nice double membranes surrounding the mitochondria. But in the hand brains, you have these electron dense regions. The cristae are not as well formed, and they are actually larger when we measure the diameter of the mitochondria. So all this is indicating you have less mitochondrial fission, more mitochondrial fusion. And not to say fission or fusion is bad, but when you skew to one way too much, it's probably not good. You know, most things are a bell-shaped curve, right? So we didn't know what was causing this. You know, it's in the brains, and this was back in 2015. I know this model is not as popular these days, but it was one of the first models, and it really, what was good about the GP120 model, mouse model, is that it did replicate much of what was seen in the brains back in the 90s, and even some of the brains that were on antiretroviral therapy. So this model expresses GP120, that's the HIV envelope protein from the GFAP promoter, that's the astrocyte promoter. And the mice show neurodegeneration, astrogliosis, and microgliosis, all which is prevalent in people with HIV encephalitis, and to a lesser extent, but still prevalent in people with neurocognitive disorders, as was shown in quite a few of our papers and others have. So we looked in actually this model and a TAP model. We didn't find these changes in the TAP model, but in the GP120 model, we basically replicated the studies that we did in the human brains, and we found reduced DNM1L. It was significant. We actually found increased mitofusin-1 in the GP120 brains compared to non-transgenic. We found reduced mitochondrial fission protein in neuronal neurons, and it was significantly reduced. We found increased levels of mitofusin-1, and we also found larger mitochondria in the GP120 mice. So this could mean that GP120 is acting directly, but it could also mean that the cells are stressed out, and so they're undergoing this compensatory mechanism to sort of save themselves and fuse the mitochondria around the soma. Still haven't completely worked that out, but I just want to keep that in mind. So all this, and there's a lot more work done with this study, and you can look at it. It was published in 2015. But we thought, if it is that mitochondrial fission is reduced because you don't have as much DNM1L in the brain, then if we replace the DNM1L gene expression, maybe we could reverse some of the pathologies in this mouse model. So we built We built a lentivirus that expresses DNM1L, and we injected it into the brain. And indeed, we were able to reverse some of the gliosis, which is, we measured GFAP expression in slices of these brains, and the GP120 mice always have lots of GFAP, but we were able to reduce that by overexpressing the DNM1L, and you can see here that was significant. And then we looked at MAP2, and you'll see this later. MAP2 is a protein that is associated with microtubules in neurons, and it is important for neuronal integrity. And you can use it to stain and determine the structural integrity of neurons. And GP120 mice definitely have less MAP2, and we were able to restore the MAP2. They have less MAP2 compared to control non-transgenic mice, but we were able to restore the MAP2 signal by overexpressing this DNM1L, and that was also significant. So this indicated that DNM1L, the reduction, may be causing this reduction in mitochondrial fission. GP120 is probably involved. And we basically, this is covering everything I said. We found the large mitochondria. We were able to reverse it by overexpressing DNM1L. So now we want to move to mitochondrial biogenesis, because we were probably a little hasty looking at dynamics, but it was very popular back then, and it had it did come into popularity because people like David Chan at California Institute of Technology, and then what's his name, George Perry, found that these processes were altered in Alzheimer's disease. And so it was kind of, it was popular. And I wrote an F-32, it got funded, and it worked out. But as I learned more and more about mitochondria, mitochondrial biogenesis is altered in many diseases, neurodegenerative diseases, and not just neurodegenerative diseases. And really, if you can't make new mitochondria, I don't know how much it matters if you can't, push them around the cell or recycle them. You definitely need to be able to respond to low energy and make new mitochondria if you're going to have a functioning neuron or any cell that needs energy. So this next study, it's really, and really the rest of this talk is focusing on mitochondrial biogenesis. not only in neurons, but also in astroglial cells. And I'll remind you why I think they are important. So now we're back into the human brain. We took brain tissues, we enriched the membrane fraction, and we did immunoblots. Maybe you'll remember, I hope you remember these transcription factors, PGC1 alpha and TFAM. These are transcription factors that facilitate mitochondrial biogenesis. PGC1 alpha is thought of as a master regulator. So during low energy, it's activated, it translocates to the nucleus and facilitates large, massive gene expression, including TFAM. But what you'll see is both of these are at lower levels in the more severe diagnoses of hand. TFAM is very much, it's a pretty robust reduction. And when we quantified this, these graphs illustrate on the y-axis the PGC1 alpha and the TFAM normalized to actin in these brain tissues. You'll see there's a significant reduction in both of these transcription factors. So it's indicating that mitochondrial biogenesis is likely disrupted overall in these brain tissues. Again, this is just a chunk of brain tissue, so you're not really knowing which cell type or that this is affected in or what it's predominant in. So we do immunohistochemistry on these brain sections, and the TFAM, it's beautiful for immunohistochemistry. So here you see this neuronal structure, and it makes sense because if you look at mitochondria in a neuron, there's a network throughout the neuron. So you'd probably find TFAM everywhere throughout the processes, and that's really what you see in the control brains. But in the hand brains, you see much lighter and less TFAM signal. And when we co-localized with MAP2, you saw basically the same thing. MAP2 is the marker for neuronal integrity. So we co-localized TFAM in red, MAP2 in green, and you see lots of yellow in the control brains. But in the hand brains, you didn't see nearly as much yellow. And we quantified all this. You did see a significant reduction in TFAM. You saw a significant reduction in TFAM by confocal microscopy, also MAP2, and then a significant reduction in the co-localization of the two proteins. So we wanted to know, because as I said, astrocytes are pretty important for getting energy into the brain and also keeping neurons functioning. We wanted to know, well, what's going on with TFAM in astroglia. What we actually found, and the one way you can find astroglia is by staying for GFAP, we found an increase in TFAM co-localizing with astroglia. So these are the control brains, and you find a little bit of red signal co-localizing with the GFAP. But in the hand brains, it's slightly increased, and you do see quite a bit more TFAM in co-localizing with these astrocytes or with GFAP. And it was a significant difference. So as we found this, we thought, I honestly thought I discovered something great, but it turns out that Enrique Cadenas up there in USC has basically shown very similar things in Alzheimer's disease and in aging rats. But it hadn't been shown in hand. Little did I know, Dan Langford was kind of working on something very similar. And we went to a meeting, maybe in Philly, I think it was Philly or Chicago. I can't remember which, but we gave almost back-to-back talks, and they were almost the same. But on one hand, it's like, you don't get to show it the first time, but it's always good to see somebody else find similar things at a meeting, because you're always questioning your work, or at least I am. So to confirm what we see in these brains as far as mitochondrial biogenesis, we looked at mitochondrial DNA copies per cell. And we found if mitochondrial biogenesis is reduced, you would think there would be less mitochondrial DNA because you have to produce more mitochondrial DNA to generate more mitochondria. And what we found was a significant reduction in mitochondrial DNA in the hand brains compared to the controls. In these same brains, they had increased levels of inflammation, or at least gene expression, mRNA for IL-1 beta, interleukin-1 beta. It's a pro-inflammatory cytokine that's been implicated in many neurodegenerative diseases. So this just sort of validated what we think was happening at the protein level. And I just wanted to kind of come back to this to remind you why we think it's important. Neurons use lots of mitochondria. They need astrocytes to bring in the glucose and then produce a lactate. So and then the neurons take up the lactate, and that's what the fuel that the mitochondria use to produce the energy. So if the astrocytes are taking this glucose, running it through glycolysis, but instead of shipping it out, they're making their own pyruvate in order to fuel mitochondria and fuel their activities, probably reactivity to HIV infection or HIV proteins or ART drugs, then there's going to be less lactate. Possibly, they're also not releasing as much glutamate or glutamine to be recycled by neurons, or maybe they're not taking up as much glutamate, which could also lead to neuronal dysfunction. We haven't worked all that out, but I'm going to show you some things that we think could sort of lead that way. I know Alicia Eugenin's doing a lot of excellent work on the glutamate area. So the next thing we did to kind of get at this and see what's going on with astrocytes is we took the brains and we isolated RNA, and we ran RNA sequencing, and we did use a bunch of, and Saranya Kanchi really helped a ton with this. We used a bunch of bioinformatics approaches to identify how the astrocyte expressed genes are modified in these brains. And this little chart here, graphic, basically shows the sort of genes that were altered that are indicated to be expressed by astrocytes. You have immune response, as you would guess, synaptic function. They do help with synaptic function a lot. Metabolic pathways, viral process, and autophagy. Really want to point out these two receptors, these are glutamate transporters, these two genes, and then DNM1L is down here. And there's another of other important genes, but I just want to point out that this is, I think it was 26 brains, and to find something this robust with just 26 brains, we were, and this is between control and hand brains, we thought, wow, astrocytes are clearly involved, and this isn't some single cell where you're pulling out the nuclei, which is very important work, but you're looking at a chunk of the brain, and you're able to identify these RNA, so whole RNA from a chunk of the brain, and you're able to identify these genes that really are consistent with what we've already seen. So we wanted to look a little deeper. We went back to the GP120 model. In this model, we added antiretroviral therapy, or at least tenofovir diproxyl fumarate. And we looked at basically mitochondrial biogenesis proteins. Something we didn't expect, PGC1 alpha was actually increased, but TFAM was decreased. DNM1L was decreased, which is something we'd shown before. I don't understand the link between PGC1 alpha and TFAM unless it's some kind of compensatory response. But importantly, TFAM is decreased, and you need that for proper mitochondrial biogenesis. And DNM1L, like I said, I pretty much always look at that just to confirm that that our models are consistent. Oh, I should have said, not only does GP120 reduce TFAM, so does tenofovir diproxyl fumarate, TFAM, and also DNM1L. We'll talk a bit more about that. So we did basically the same work, co-localizing TFAM with MAP2, and we found reductions not only by GP120, but also tenofovir diproxyl fumarate. The combination reduced the TFAM even more. The same with the MAP2, although there wasn't an additional reduction with tenofovir diproxyl fumarate, but there was an additive or subtractive, I guess, a reduction with tenofovir and GP120 with TFAM and MAP2 co-localization. So then we looked at astrocytes, and we found some very strange things. But basically, GP120, you always see more GFAP in these brains. Tenofovir induced astrogliosis. again, I thought I found something great. Turns out this study out of Africa, they actually used this model, maybe not this model, but they gave mice tenofovir and they'd shown the same thing as far as it induces astrogliosis. But it also increased TFAM in astrocytes. I wish these pictures were bigger, but all these astrocytes that I picked out here are chock full of TFAM. Like right here, you can, I don't know if you can, but that's red. That's astrocytes just chock full of TFAM. So more so than we saw in the brain, which makes sense. This is a model, plus it's also expressing GP120 from astrocytes. But I think it validates that these astrocytes are capable of ramping up mitochondrial biogenesis, which means they're probably not giving the energy that would normally be given to neurons. This is sort of a tangent, but it's the same study. I just want to put it out there because I think it's really neat. Tenofovir reduced microgliosis as measured by IBA1. So GP120 mice, you get lots of IBA1. IBA1 is a marker for microglia. But when you treat the wild type and the GP120 mice, you have less IBA1 signal. And we found that over and over. Not only that, the tenofovir reduced IL-1 beta and TNF-alpha mRNA in these mice. Don't fully understand it, but these next couple studies will try to get at it. We just haven't been able to really dig into that, what's happening. So reduced mitochondrial biogenesis, inverse relationship between TFAM and HAM. So less mitochondrial biogenesis, more cognitive impairment. Tenofovir induced astrogliosis. Tenofovir blocked microgliosis. So because the astrocytes were increasing this mitochondrial biogenesis, we were wondering, are they, good targets to restore mitochondrial function in neurons and reduce inflammation, which we think they are. So the first thing we went into a dish. You can see a little astrocyte there. We have astrocytes in a dish, and we treated them with IL-1 beta and tenofovir. And what we found was tenofovir, and we measured IL-1 beta as well. So we measured pro-inflammatory mRNA expression, IL-1 beta and TNF-alpha. And on the y-axis, you'll see IL-1 beta levels normalized to actin, and also TNF-alpha levels. And what we found in astrocytes was that IL-1 beta, is well known, induced IL-1 beta and TNF-alpha mRNA, but tenofovir significantly reduced those levels. When we went to microglia in a dish, tenofovir did not have that effect. And really, we were trying to see how tenofovir reduced inflammation in the mouse brains. I just showed you. According to this, it could be by astroglia, even though we saw less microgliosis in those brains. Again, I don't have a lot of answers for this, but it's what we found, so I wanted to show it. And the effect on astroglia, because we looked at multiple donors, it didn't reduce inflammation in all the astroglia, but as I'll show you in a minute, in all the astroglia, it did induce metabolic activity. So next, we were wondering, okay, does tenofovir induce metabolic activity in these astrocytes? And we found that it does. We used the seahorse respirometer to measure oxygen consumption rate. Sorry about that. And we treated with increasing doses of tenofovir. We saw spare respiratory capacity was increased, and the extracellular acidification rate. So tenofovir does increase metabolic activity in the astrocytes. So next, we wanted to, I'm going to have to start talking a little faster. I apologize. The next idea is to figure out, okay, can we block this inflammation or this activity, metabolic and inflammation in astrocytes, and will this protect neurons? As you may know, the literature is mounting up that, and we know that people with HIV use higher levels of cannabis, and there's some evidence that cannabis reduces inflammation. And in people with HIV, not everybody, but in people with HIV, there's some reduction in there may be a reduction in neuronal dysfunction. So we use this model. We have astroglia in a dish. We treat it with this cannabinoid receptor agonist Win, and then IL-1 beta. Then we measured the respirometry or metabolic activity. We looked at some RNA, some protein, and immunohistochemistry. I don't think I will show all of that, but I think we get to the point as far as just like with tenofovir, when you treat astrocytes with this inflammatory cytokine, you get increased metabolic activity, oxygen consumption rate. If you treat with Win first, this cannabinoid receptor agonist, you're able to block this increase in oxygen consumption rate. So you're able to block their metabolic activity, which, remember, if you do that, you may be allowing them to produce more lactate for neurons. You see reduced levels of ATP with increased levels of IL-1 beta. We think that's just because they're using up the ATP for gene expression and changing the morphology and all sorts of things. But if you pre-treat with Win, you increase ATP levels, which probably you're just settling them down so they're not using up ATP as fast as they're making it. So then we wanted to see the effects on inflammation. We treated with increasing doses of IL-1 beta. This is nothing new. You get increasing levels of IL-6 gene expression, but when you pre-treat with Win, you're able to block the IL-1 beta induced increases in IL-6. And that's actually been shown in some mouse and rat astrocytes. So that also wasn't very novel. But we wanted to figure out how the Win is acting. It could have been through CB1, PPAR alpha, PPAR gamma. So we use these inhibitors and knockdown, and it's all in this paper. But what we found was the only thing that was able to block the activity of Win to reduce IL-1 beta induced inflammation was this GW641, which is 6471, which is a PPAR alpha inhibitor or antagonist. So we think that PPAR alpha is mediating the role of the cannabinoid receptor agonist to block inflammation in these astrocytes. Okay, so the next question was, what does this mean for neurons? So we have astrocytes growing in a dish. We have neurons growing in a dish. We treat the astrocytes with IL-1 beta and Win, alone or together. And then we take the conditioned media and we add that to neuronal cultures. And then we measure the mitochondrial. We measure TFAM. And that's what this will show you. So these are all treated with conditioned media from astrocytes. You have the control, so that's just conditioned media with untreated astrocytes with IL-1 beta treated, and then Win alone, then Win plus IL-1 beta. The TFAM signal is in red, and you see there's a reduction in how much TFAM is in these neurons. There's also a reduction in the MAP2 signal. With Win, you don't have a difference, but with Win plus IL-1 beta, you were able to restore the TFAM in the neurons. And this was all shown again here when we quantified the signal for TFAM in MAP2. So maybe, oh, I thought I had a little marijuana leaf. Maybe you're able to reduce astroglial reactivity and increase mitochondrial function in neurons with cannabinoid receptor agonists. So again, the reason we think this is important is astrocytes, they're more than just the glue of the brain. They're actually bringing across metabolic substrate. They are, in normal conditions, they are producing lactate or metabolic substrate for neurons. They're regulating glutamate and glutamine in the extracellular space. And if this is altered, then a lot of these processes probably aren't functioning correctly. They may also be releasing a neurotoxin instead of just withholding neurotrophic factors. And maybe cannabinoid receptor agonists can help with this. So what we really want to do is identify the specific pathways in astroglia that are mediating this neurotoxicity. Maybe it could be addition of toxins, production of toxins, or lack of neurotrophins. We want to look at lactate and glutamate transporters. And then really we want to investigate the relative role of glycolysis versus oxygen, oxidative phosphorylation in astrocytes. And we'd really like to get to where we're using patient-specific mechanisms, using patient-specific models to identify patient-specific mechanisms and test therapeutic strategies. So there's a ton of people that helped with this. Obviously, I couldn't have done all this and couldn't do any of this without support from the NIH. So thank you to everyone. And yeah, thanks for listening. I think we got 10 minutes. Unless y'all want to hear more, I do have more.
Thank you, Jerel. I don't see any questions in the chat. So I'm just going to invite anybody who has a question to unmute themselves and ask the question directly.
I will show y'all more if y'all don't have questions.
Okay, I have a question. Go ahead. Adam, thank you so much. I want to say what a wonderful speaker you have become over the years, and I really like to hear you. So I was looking at your mitochondria, and in addition to have increased size, they also have the crystal kind of, you know, the architecture doesn't look very good. What do you make out of it? Do you see a role, let's say, for reactive oxygen species, you know, oxidative stress? How would you place oxidative stress in the phenomenon that you are looking at?
Well, I think it's probably prevalent if for no other reason. I know people have shown that antiretroviral therapy can affect or help produce oxidative stress, and then HIV proteins can, but also these HIV proteins can also reduce, if you reduce mitochondrial fission, then you're in turn reducing mitophagy, which means when the mitochondria are damaged, they aren't getting recycled at the rate that they probably should. So you're probably having a buildup of damaged mitochondria, which that means they're not going to be able to. And if you have reduced mitochondrial biogenesis, you can't produce the proteins for the electron transport chain, or, you know, produce the cristae and all the proteins that are needed there. So I think it's probably not just one thing, but I think, yeah, oxidative stress, it probably plays a big role, because that's going to also damage proteins. And you probably don't have the antioxidants being produced at a rate that they should. So that's kind of why I think if you know, metabolism is a thread through metabolic disorder, it's a thread through many neurodegenerative diseases. People with diabetes have a higher prevalence of neurodegenerative diseases. So I think, and I didn't show all the data, we definitely have other things that make me think this, that one sort of way a general way to address this would be to address metabolic issues and what people are eating, how much they're exercising, how they're breaking down certain things, you know, what's getting to the brain. The vasculature is super important in all of this. And I think if we could do that, a lot of the oxidative stress and mitochondrial damage would fix itself in some ways. And I think astrocytes are going to have to be involved.
Yeah, that's interesting, because usually you hear of PGC1 alpha in fat tissue, in brown fat and white fat, you know, the balance is really metabolic indeed.
Thank you, Cecilia.
I have a question. Very nice work. The extent to which these mitochondrial are morphologically modified, right? The fission and fusion looks quite impaired. Have you looked directly at the metabolic substrates that are used once the mitochondrial get into that state?
Well, I can say we're doing that work, and we're hoping to get funding to do more of that work. That's a great question. I'm not going to give away too much. There's too many good scientists here, and I'm too desperate for funding right now. So I can't do that in papers. But yes, we are. And I think that's very important, because yeah, you can change the pathways that are being used, and what that will do to the mitochondria. Will it restore their integrity? I don't know that yet. But I have a pretty good hunch that we can alter the health of neurons by, there's other work out there that shows metabolic substrate is very important for other neurodegenerative diseases. So yes, I think it will make a big difference. I don't have an answer for you now, though.
Okay, no, that's great. Thank you. I figured since you're there with Diane and she's done some of this metabolic work that you're probably going down that road.
So Alan McCutcheon has a question, wants to know more about WIN. Alan, do you want to
Sure, just, I wondered what kind of cannabinoid and what receptors it agonizes and whether it's anti-inflammatory in other settings and so on.
Yeah, that's a good question. So when it's a synthetic cannabinoid, it's not, based on what I've seen, it's not really promising as a therapeutic, because what it does to cells, it is anti-inflammatory. They used it in, I'm pretty sure, models for Alzheimer's disease, but several mouse models and rodent models for neurodegenerative diseases, and it is consistently anti-inflammatory, but it does something to the endoplasmic reticulum, and it does something to induces this vacuolization of cells, which I actually show pictures of it in my in a paper, and we mention it, but the reviewers kind of let me slide on it. But I don't think it's a great drug. I don't know though. I mean, I'm not at that level, and I haven't tried it. But based on what I've seen at the cellular level, the levels that we had to use to be anti-inflammatory were also toxic in some ways, but at a different in a different process. But as far as the receptors, it activates the same receptors that many cannabinoids like CBD activates, CB1, CB2, PPR alpha, PPR gamma. I'm not sure about like GPR 55, but I know it has been shown to activate those receptors. We looked at all of those in our study, but we found that PPR alpha was the proponent, or at least the mediator of the anti-inflammatory activity in astrocytes. I would like to try other cannabinoids, though, honestly, some that aren't so directly affecting the vacuolization of cells, because it really looks bad in vitro models. We looked at an electron microscope, and it was not good. So, but it did what we wanted it to do, and it actually did protect the neurons, as we showed. But I think prolonged use, more studies need to be done. I'll say that.
Hey, Adam, this is Chris. Hi. I want to second what Cecilia said. Very nice presentation, and you cover a lot of ground, and very clear and informative. So thank you. I have a question about, maybe I missed it, what you said. Did you compare by any chance THC to CBD in terms of mitochondrial protection?
I have not done that yet. I have not. It's in, as I was kind of alluding to, I think that's where we need to go, and maybe even some of the other cannabinoids, the lesser well-studied. I think there's something like 16 and different cannabinoids, but no, I haven't done that, but it's definitely a future direction.
All right, thank you.
Thank you.
I have, we're just about at the hour, but I have one general question that maybe you will share with us some more about what you mentioned early on. You mentioned a clinical trial that either beginning or about to begin with polyneuropathy, distal sensory polyneuropathy, where it sounded like you were targeting mitochondria with something. Would you mind sharing a little bit about that trial with us? Yeah,
for sure. So there's a, I can't remember if I have it on here or not, but so there's a, I'm across the hall from these folks that study, I don't have it. They studied diabetic neuropathy. And what they found was this anti-muscarinic, a muscarinic receptor antagonist induces mitochondrial biogenesis in sensory neurons. And sensory neurons, they're pulled back in this neuropathy. And what they think is this, whenever they're pulled back, they're sort of just firing aberrantly. But they have this drug, it's called parenzapine, and we published this. We just put it out recently. When you, in mice, it was tat mice, and they've shown it in other models too, I think a rat model as well, diabetic neuropathy model. It basically induces mitochondrial biogenesis, it restores electron transport chain proteins, and it reduces indices of neuropathy. The allodynia, the von Frey filament, the whole reactivity there, I think temperature sensitivity. And so, yeah, it's anti-muscarinic, and I've worked with Ron Ellis. He was on the paper, and I know that I didn't go into the clinical trial part, obviously, because I'm not a clinician, but they got a good score, and I think it's probably going to be funded soon. And they're basically testing in HIV patients. It's a topical ointment that you rub, and they're going to take biopsy so that they can measure the neurites, the sensory neurites that are basically innervating the skin. And then they're going to, I think they'll measure some mitochondrial markers as well. So anyway, yeah, it's mitochondrial biogenesis. It's in the periphery, and yeah, hopefully it works out. I wish the brain was so easy to get to, but as you know, it's not.
Thank you. It sounds like an exciting study. I'm going to look forward to hearing about it. Let's just close by thanking you again for a very interesting and stimulating talk, and to all participants for discussion. Next week, we have 2 trainees on board. Lindsay Festa will be talking about lysosomal deacidification by the antiretroviral bictegravir inhibits oligodendrocyte maturation. And Sylvia Torces Deval both from Miami Miller School of Medicine. She will be talking about targeting occludin in human brain pericytes, implications for HIV-1 infection and immune response. So we'll see everybody next week for 2 trainee talks.
Thank you all.
Have a good day, everybody. Thanks again.